I'm going to talk about the context of this project. At the beginning was I was creating a function. There was a translation of the web-based statistics that was developed by Zaleski like 11 years ago, and is widely used in neuroscience, neuroimaging community. And there was an issue in one of the projects. They want in my lab they wanted to use for these Prairie balls, and they wanted to extract a circuit in the brain, so what is a cluster of connections. But the problem is it was a longitudinal design, and some of the participants didn't finish all of the scans, so that was a problem with the original toolbox. And using linear mixed effect is more efficient to deal with missing values. And the one particular funny thing of these Prairie balls is they have a very interesting sexual activities. And for example, in the first season of Rick and Morty, they used a portion of the hormones of those balls to try to Morty conquer Jessica. And they have well, just to say one thing. For example, if these balls have when they mate together, they copulate, and if they stay together, they create a couple. But if they mate but you separate it in different rooms, they are not a couple. So it's a very funny behavior, similar to humans. So well, this is the framework of the network-based statistics. So we wanted to have a relationship of some variables of interest that they can be a clinic, cognition, genetics, and we wanted to relate to a 3D dimension of brain networks in a third dimension domain of subjects. If we use like a normal multiple comparison approach, Bonferroni of FDR, is very, very conservative, and you lost a lot of, yeah, you are going to have also a loss. You will control the false positive, but you are going to lose a lot of power. So what the network-based statistics does is find like cluster of connections, and these connections you can jump to one node to the other just with these connections. But for example, you cannot jump with this cluster to the other cluster. So you take an a priori threshold, you create these clusters, and in the other side, what you are doing, you start to do this permutation of your own data. You create a null distribution, and you extract the maximum cluster for its permutation. So that means you are going to test how is the maximum size you can get by chance. By chance, by a random approximation. And finally, you test the size of your clusters inside of this null distribution, and eventually you have these p-values. And yeah, we created last year. It was downloaded, but a lot of times. Maybe it's not necessarily 6,000 people, because so many people start to download it many times. But yeah, I start to receive mails and with inquiries, and it was very nice to receive feedback from the community. It was very, I think one of the best things I did in my PhD. And one of the main problems that the user said is that it was very, very slow. For example, for these small brains, few regional interest in the brain, there was not a big problem. But when you start with work with large brains with many regional interests, yeah, the computation, it takes hours, sometimes days. So that was the start of this implementation in Python. Now it's Luba.
Hello. Hi, thank you, Zeus. Sorry, I had to change my spot quickly. So yes, I got involved in this project because I wanted to contribute from the technical side. So the biggest challenge was to actually improve the performance in terms of execution time. It's not just that our code is slower. It is that Python has some kind of additional features that R doesn't have, and we have explored it. So if you can move to the next slide. Yes, so first we explored the stats models that I guess everyone has heard of the framework, and it allows actually a smoother transition to Python for someone that doesn't know Python, from R to Python. And there is this API that it's very R-like. However, in the next slide, we show that it is possible to pretty quickly compute linear regression if we are given one column of endogenous and some exogenous relation. And it's, I mean, relatively pretty quick. However, in the next slide, yes, if we run a linear regression across all the columns that we saw, matrices that are, it takes 1 second. Is this good? Most probably not. It's not beating R at all. So we had to go into another direction. We had to find a way to use an advantage of matrices and operations on matrices. So next slide. Yes, so what is showing, okay, sorry, prior to this, we actually wanted to know if we can give a multiple inputs as an endogenous variable, but unfortunately it's not possible. This is what we see from the stats model documentation, official one. So the next slide actually is showing and proving to you that there is an value error. We cannot fix it. I mean, it's a hackathon. We have to think smart. So what we did is that we developed a Python, skill-learn version. I'm calling it skill-learn because it's relying on NumPy. So next slide is showing our version of what you saw previously that ordinary least square is doing. However, what's the disadvantage of this part is that we have to pre-process the data. It's not an API that it's very R-friendly. What it is, another disadvantage is that we have to actually compute P and T values by ourselves. But this is giving us a good advantage of utilizing broadcasting and actually operation on matrices. I wouldn't go through code, definitely. So the next part is showing you that we could achieve from 1.24 seconds to 56 milliseconds. We could fit a linear model. Now, the next part is actually permutations. And this is, yeah, so no. Yeah, you can go to the next slide. It's fine. Thank you. So so this is just within one permutation, like an initial model that we need to compute. Before going and explaining the results, I just want to emphasize that NumPy as a backend behind has a lot of disadvantages for statistics. As we can see, pre-processing categorical columns is necessary. We need to do this. Is there a formula that we can provide like in R? No. Is there a way to compute P and T values? I mean, there is now with a bit of Google and actually going to the basic math. But the biggest advantage was that we could use in NumPy matrix operations. And this is what we achieved in Python. So the next slide is actually showing us what are the results at the end. And this is Walid going to proceed. Thank you.
Thank you. I think next slide. Yeah, so my background actually is mainly in genetics. So, and I wasn't sure actually if I could help actually in this project. So, and I joined, but I was glad actually there is some, at least some part that I could contribute to. So I actually contributed to benchmark the tools actually, and then at the beginning, we wanted actually to understand, we had two data sets with different, size, the first one is around 48 rows and then over 300 columns. And there's such a small data set. And we looked at the permutation, compared to the time, needed actually to finish the job in terms of permutations, which would give us actually the efficiency in terms of time. So, and it clearly actually, if you have, if we compare the first two data sets, actually, example 1 and example 2, you see actually that the time exponentially increase when it comes to, increase in permutations. The second part was the most important part actually, because it was mentioned before that in terms of time efficiency was a challenge. So and we wanted to see the implementation of R and Python actually, whether we do have a big difference and we could actually save more time, which is the most critical part here. And as you can see, actually, there's quite a striking difference actually in terms of time. We use the same almost the same machine actually for this one for the testing the implementation, and it took what was actually achieved by Python like within seconds, I guess around 50 seconds or around that number. It took like over 400 seconds for R to finish the job, which is I'm not sure how many fold of reduction actually in terms of time. I think I'm getting back to Zeus actually for concluding actually this.
Luba, I think you are the expert in this part.
Sure. I think I just saw that I made some grammar issues here. Yes, as everyone knows, I just want to give some kind of brief introduction. I didn't know R prior to this. I knew some basics, so I learned a lot from, let's say, copying the code or translating into Python. And I saw that R is very rich with statistical libraries, computing it quickly for some, not big data sets. I'm working with a big data set, so I have some kind of experience in parallelizing and multiprocessing. But here, it's not just that Python proved faster. The point was actually to discover that there is where there is advantage, there is always disadvantage. And we actually want to continue working on this and make a better API, because I cannot say aloud that we saw some flaws with stats model, definitely no, but we could improve some things. So this is our next goal. And yes, we have to still work on improving some parts of code that can work better, because we also use pandas, and when we go to packaging, we want to reduce number of dependencies. Therefore, our conclusion is that if you're very comfortable with Python, yes, proceed. Otherwise, R is efficient. But in this particular case, as Zeus said, he was contacted by a lot of people, and this is where a need was created to build something, let's say, efficient and maybe not impressive for sure, but efficient that people can use. So yes, this is what we would love to conclude. Thank you. And thanks for my colleagues for contributing. I mean, Zeus is the one who actually proposed the idea.
This is great.
He was working on this package.
Congratulations! That's really a great project, I thought. Guess let's see. Are there questions, comments?
I'm just wondering if you have plans for distributing this on like NeuroHackademy or anything like that.
Yeah, definitely, with a lot of help from other team members, because I cannot work alone on this. I mean, I'm not intending to, but I mean, this is a theoretical part more that we can improve and continue with this. But definitely, yes, that's our next step.
I guess another question I have is whether some of the things that you found out could go into stats models itself. Right? It seems like you've figured out a way to calculate these things, including p-values and t-values on multivariate data sets.
Exactly, multivariate.
Improve stats models and make that faster, right? Not have that error.
Yeah, I mean, this is a possible way. Maybe they did it for some reason that we couldn't figure out in 4 days. I'm not completely sure, but most probably we have to investigate stats model more. Personally, myself, I don't use stats model for deployment and or even analysis. I used it in a very limited time and in a very limited occasions. So I'm just stick to NumPy. But yeah, I think this would be this can be a good way to prove that maybe we can improve this. But yeah, it requires a bit of research.
Great. Other questions, comments? Okay, let's move on then to connectivity ML. Does one of you have the slides to share?
Hi, sorry, my computer's being a little slow here. I'm just pulling the presentation up now.
Great.
Oh, these are all wrong. Sorry, it's being super slow. Okay, and here we go. Sorry for the delay. Can you see my screen?
Yes.
Okay, great. Can I make the slides move? Yes, okay. So we wanted to ask a question about looking at individual differences in resting state connectivity, and if that had any relationship to to individual differences in things like cognition or demographics or other features. And it's when you have a whole network, it's kind of how do you get your head around that? How can you use that data? How can you interpret it? So one of the things we're also interested in is which particular edges might be more predictive for some of these outcomes that we're interested in. We use data from the Human Connectome project. We have resting state data for 1,003 participants. One kind of interesting feature of this data is that there was connectivity matrices already available, but the way that they parcellated the data was using a spatial ICA. So the way that turns out is that the parcels aren't always anatomical. They can be non-contiguous and they can be overlapping. They also released this with several different parcellations, so between 15 parcels and 300 based on components. And so that's something that you'll see in our presentation. We're referring to the different number of components, which parcellation set we used. But like we're used to seeing for resting state data, we had time series correlations for each pair of components or each pair of parcels. And of course, that's what we interpreted as edge weights in our networks, and that's what we used as predictors. And now I'll pass it over to Tao.
Thank you, Priya. So in this project, we predicted a subject gender using a random forest classifier, which is basically a large number of decision trees operating together as an ensemble. A great option with the random forest classifier is the ability to measure features importance. Here, we decided to look at SHAP values, which is an alternative to the standard feature importance, and is implemented in a Python package that works for tree-based models and scikit-learn. How comfortable. SHAP values are based on the magnitude of feature attributions calculated by summing the absolute shapely values per features across the data, where the features with the large absolute shapely values are important. Next slide, please, Priya.
Oops.
Okay, thank you. So now feature importance is useful, but contains no information beyond the importance. And with SHAP values, we can dive into more details by looking at the SHAP value, SHAP summary plot. That's the plot we see here to the right. And it tells us much more. First, the feature importance that is represented as the variable's rank in descending order, basically meaning the top one, 129, in this example is the most important. We can also learn more about the impact of each features on the prediction. So the horizontal location shows whether the effect of a feature is associated with higher or lower prediction. And finally, we can also look at original values, whether, so that is basically where color shows us whether the value is high in red or low in blue for that observation. Great. Next slide, please. So now, beyond looking at our average result, we can also drill down to the specific subject result, like we see here for the ACP subject 101107. First, it's a lot of figures going on here. First, on the top, we can see the SHAP force plot showing us decomposed single prediction, where the output value is the prediction for that subject that we evaluated, the 0.41. The base value is the value that would be predicted if we did not know any features, basically the main prediction. And then in red and blue, we can see which features, which are edges between two ICA components, push the prediction higher, meaning a female in red, and those pushing the prediction lower, meaning male in blue. Now, then we can take it to the next level, as you see on the bottom, and show these results with the most important ICA components on the brain itself, as you can see to the right, or on a brain free graph to get just a better understanding of the results in terms of parcellations and maybe even anatomy that is a bit difficult with these parcellations. Thank you. Go ahead, Debbie.
Great. Thank you, Tal. So then we were interested to see whether the SHAP analysis would be stable, and we evaluated that looking at a 5-fold cross-validation. We did that for, as Priya was mentioning, there are different ICAs with different numbers of components. So we did that for the 15 and the 300 to look at the both sides of the extreme. And here you can see, so this is for the ICA with the 15 components. And the plots are for the mean SHAP value at the top is the feature, the edge with the most, that contributed the most to the prediction. And then it's in descending order of magnitude of importance to predictions overall to all participants. And here you can see that the edge 84 actually came out on top for all 5 of the folds for the ICA 15, and also the edge 84 has a much greater magnitude compared to the second most important one in most of the plots here at least. And also similar edges are found in like the top 4 or 5, 66 or 13 or 74. So from this we might conclude it is reasonably stable. Next slide, Priya, please. And here you see the results now if we look at 300 ICA components. And you can see, first of all, that the top one is very different. And also out of the other 20 important ones, they vary quite a lot. And the first one also doesn't stand out as much anymore as having a much greater magnitude in all the 5 folds. And for example, I looked at the top 8 for all of them, so 40 in total, and there was only 5 that were in duplicate. So we think that there are potential reasons for why it is less stable with 300 components. Could be that perhaps it's more granular. With 300, you might also have, as Priya was saying, some might overlap some of the regions on the brain. Perhaps there are 2 components that are part of a network and a functional network in the brain, and maybe you have very similar edges from one versus the other component going out, and maybe sometimes one wins out, sometimes the other. Those were just hypotheses we had for why it is less stable, but this is something we'd want to explore more if we had more time. Next slide, please. Yeah, so we didn't have the time, but there is a paper that also looked at gender prediction using the Human Connectome project from 2018. And they used a subset of our data, so about 80% of the participants that we included. They had a similar accuracy to the one that we were able to achieve. And they were able to explore the existing functional networks, and where they had the most important features were from the default mode network, frontal-parietal network, and sensory motor networks. And they had a conclusion that those might, in the past, were implicated in social functioning, and that might be a potential reason for why those were helpful to predict gender. And those were the things that we also would want to do if we had more time.
So oftentimes, as a person who's working with this kinds of data set for the first time, we have to make a lot of decisions because of the sheer amount of data that is available. So a few decisions we had to make was where, as Priya was saying earlier, there were several different parcellations using spatial ICA. There were 15, 25, 50, 100, 200, and 300. Which one of them would give us better performance? That is something which we had to consider. The data release also had 2 kinds of connectivity matrices. One was the PSS correlation coefficient. The other one was partial correlation, which actually controlled for the other time series or the other nodes. We also wanted to know which model to use. So as we showed the results for random forest, but would support vector machines be better? That's something which we also wanted to explore. And finally, once we choose our model, there are several hyperparameters as well, which we wanted to optimize. So that's what we plan to do. Can you go to the next slide? Yeah. So as gender as our outcome variable to predict, we built a combination of these models. So basically what we did was we used a nested cross-validation loop in which the inner cross-validation loop was essentially trying to optimize the parameters, and the outer cross-validation loop was trying to predict accuracy. So as you can see, the results are shown on the figure on the right-hand side. The first row is essentially for support vector machines, and the second row is for random forests. And the first column is for correlation, which is netmax 1, and the second column is partial correlation for netmax 2. So as you can see, a few general trends emerge. So the more the ICA components in your parcellation, that is the number of components in your parcellation, the better the performance is. Another conclusion was that the partial correlation, as you can see, was performing much better than using Pearson's correlation coefficient, which was not surprising. The third is support vector machines actually performed much better than random forests. In fact, we were actually able to get a performance of 95% accuracy in predicting gender when we used like 100 IC components for support vector machines, using support vector machines. And also parameter optimization in our case was not that helpful. In some cases it was helpful, but it was not that helpful because again, we did not try to optimize all the parameters, but we just wanted to optimize a subset of parameters because obviously the computational complexity explodes greatly if we consider all combinations of hyperparameters. Go ahead, Kai.
Thanks, Pradyumna. So to summarize, our analysis has suggested that several age weighting features derived from the ICA are important for predicting the individual differences in the Human Connectome project's resting state fMRI data. And our analysis also has some limitations. And for example, when we are exploring increasing amounts of the independent components, of course, the accuracy is increasing, but it gets harder and harder to interpret and visualize this data and get some meaningful interpretations out of it. As well as the stability also decreased, as is suggested earlier in the SHAP analysis. And there is also a point that has been pointed out by Debbie in the literature that gender can account for a huge amount of the difference. So our analysis didn't account for gender effects, as well as the whole datasets of human connectome is actually not consistently processed through a pipeline. There are subsets of the subjects going different preprocessing. So we might also take into account of that as well. So in the future, as has been described in Pradyumna's slide, there are a lot to optimize for model selections and performance, and that's on a modeling perspective. And from a data perspective, we might be able to explore fMRI data from the HCP projects with different approaches of parcellations, other than the FSL melodic, which use a spatial ICA approach. So that comes to the end of our presentation, and we would just like to thanks Noah, Ariel, and Eric for the amazing assistance to be able to set our project up and running so quick. And thanks to all the organizers for NeuroHackademy and NIMH and eScience Institute. We really enjoy using these projects to quickly implementing some new ideas and visualize things and just learn. It's very enjoyable. Thank you.
That's great. Congratulations. That was fantastic. What questions, comments do people have? I guess just one comment here to make is that it looks like you beat the state of the art in gender prediction in terms of just performance by a pretty large margin relative to that older paper. So that's impressive. So that's cool. Other questions? NeuroHackademy, I see your hand up. Yeah.
Sorry.
Hello. Yes. Impressive work. Yeah, even though it's not my domain of knowledge, I was just trying to understand why SVM is particularly beating random forest. Is it because you were using linear kernel or something else?
I think the SVM implementation which we used was a linear kernel. So we just used a linear SVM. To be honest, it's really hard to say why SVM is performing better than random forest on this particular data set. Again, probably because, I mean, the only thing which I can think of is because SVM uses a hyperplane to maximally separate the data points in space. It just happens that in this particular data set, like linear SVM, when we optimize the parameters, it's just performing better, I guess. Does anybody else have anything else to add as to why?
That was...
Yeah, I would also guess that, oh, sorry, Priya, go ahead.
No, go ahead.
I would also guess that the random forest overfits pretty badly without pretty substantial tuning of hyperparameters. It has there are a lot of hyperparameters you can set for random forest. So it's likely it's overfitting to the training data without a lot of additional exploration.
Thank you.
I can confirm we've seen that overfitting with random forest.
Yeah. No, you had another comment question?
Yeah, I was just going to say that this is really cool considering so I've looked a little bit at gender in the Human Connectome project, mostly in data in the visual cortex, and we found it very hard to distinguish. Of course, we weren't explicitly training, but nonetheless, this is very cool. This worked as well as it did.
Svi, I saw your hand also.
Yeah, I was just wondering whether you considered the HCP, I think, as you said in one of the stand-up meetings, one of the previous days, is so rich that it's kind of tempting to just say, why not throw in there some anatomical statistics or, so many other things that we could calculate from from those scans, and I was just wondering whether you consider taking that in that direction and seeing if you can hit that 99% region.
I can tell you offhand. Oh, sorry.
No, go ahead. Go ahead.
I was just gonna say, I can tell you offhand that the male brains in the human connector project are about 40% larger than the female brains. If you just look at cortical surface area, but as soon as you scale the brains, it's like you look at a single functional area and you scale it by the cortical surface area, it's like identical across genders. So I'd be curious to see how that did too.
I actually did the comparison recently with for a few hundreds of subjects, not from the HCP actually, just a few hundreds of subjects from our lab, using only the anatomical statistics. So I guess, no, that should be normalized. The anatomical statistics outputted by FreeSurfer, by the regional pipeline. And we actually got good results. I never got to really kind of optimizing that pipeline all the end, but it was well over 80%. It was kind of surprising. So I guess that there's a lot of information there as well, it seems.
Yeah, so just from my perspective, I'm definitely more interested in interpretability than prediction. So that's something that felt important to me. And I felt like, okay, if we're a little bit disciplined about what variables we're putting in, the edge weights are something we could potentially interpret and say, okay, maybe this particular connection between these particular regions, if they were regions, would have some kind of meaning. With the anatomical, depending on what you were looking at, for gender, sure, for biological sex, there could be interesting differences. For me, I'm coming from more of a psychology place. So what I'm hoping to do is take the approach that we did here and use that to dig into some of the cognitive variables. And for those, I wouldn't imagine that the anatomical variables would be helpful, although there are definitely other variables in the data set that could be helpful. And then, yeah, you get into the question of, should you just throw everything in there that might help? Or what does it mean if you're putting like edges in with like task data? Does that even make sense in a model together? And it might. So again, yes, big data, but theory first. That's how I feel.
Great. Cool. Let's move on to the next project presentation, which is, I've got to find it, feature encoding.
Great. I will go ahead and share my screen. And it would help if somebody can confirm if they see my screen.
I can see your screen.
Awesome.
And I can see your slides.
Okay, awesome. With that, I will kick off the first slides. Hi, we're the feature encoding group. And our title is Feature Encoding with Facebook's Slow Fast Algorithm. Our project goals were pretty simple. We were very practical, where we all wanted to have our hands get dirty. We wanted a hands-on experience on deep learning, using state-of-the-art video extraction algorithms. So we went for Facebook's slow fast algorithm. And from that, because we're neuroscientists or interested in predicting the brain, we decided to build a feature encoding model with the extracted annotations. So here's a little figure to show you the process. We first started by selecting our data, and then we fed it into the slow fast algorithm using pre-trained weights. And then we reduced the dimensionality of those weights by PCA. And with the dimensionality reduced weights, we fed it into a lasso regression model to predict the brain. And our teammates are going to explain the steps one by one.
Yeah, so we used the 7 Tesla functional MRI data from the Human Connectome project, in which participants watched short blocks of videos during 4 runs of acquisition. And the videos were selected from like various Hollywood films like Inception and Home Alone, as well as a few independent films. So on the right, we're just showing a few example frames from an independent film about a flower that, as a side note, actually took me on a unexpected journey personally. So 11 out of 10 recommend. But anyway, one cool feature of these data is that they had already been pre-processed using the HCP pre-processing pipeline. So for example, all the data had been normalized to the standard MNI template and were denoised using the ICA fix algorithm. And so the full data set contained a complete data from 184 participants, but due to some storage and transfer limitations, here we're only analyzing data from 30 participants.
All right, so we had both video and fMRI data. So for the video data, we have to do some pre-processing in terms of resampling it to manageable resolution for our project. Then we split it up into frames using the FMPG tool. And then we took the frames belonging to each video segment and put them into separate folders in order to be able to process them independently. So as you might have seen, this dataset includes 15-minute videos and there are various segments. So we just made separate videos from that. As for the fMRI data, we did some gray matter masking and resampled this to 3 millimeters in order to be able to process it more efficiently using our resources, which actually were provided by Ariel. So thank you, Ariel. And then we proceeded with the next step.
Yes, so we have used these slow fast networks for video recognition, which involves this slow and this high pathway. And this slow pathway operates at a low frame rate to detect better spatial content, while this high pathway has high resolution. This means more frames per second, but uses less channels, which makes this lighter. Also, there are lateral connections that fuse pathways, sharing information between these two paths. In our work, we used the pre-trained weights as it was not possible to retrain the network from scratch from a ResNet 50 architecture. So each sample of this model corresponded with a frame of the video, and the targets were 400 features. And then this information has to be summarized in a way. So we take the average in order to match the fMRI data, which have a 100 resolution.
All right, so following slow fast feature extractions, we then use principal components analysis to really get a sense of how these features were identified from our stimuli and how they were clustering. And so we found about 63 principal components that we used in our final analysis. And just to visualize these clusters, we used word clouds. You can see that word cloud from principal component 1 on the right there. And honestly, it was pretty difficult to interpret how these features were clustering based on the principal components. For example, in principal component 1, we see things like, okay, dyeing your hair or biking through the snow and even throwing an axe. So it's not really like you can say, okay, this is clearly a cluster about, sports actions, or maybe something about whatever axe, like axe throwing is related to. But what is interesting is that we looked at a couple more of these principal components, making our way down to like the lower orders, and that as we start looking at these lower PCs, they seem to become a bit more interpretable, and we kind of see some more specific actions. So for example, this is the word cloud from principal component 5, and we see things like almost sports action related. So things like American football or even dodgeball. And so that this may be a little bit more sensible and possibly related to like the lower variance for these principal components.
Yes, with the reduced dimension video features, we move on to predict voxel-wise brain response based on the principal components. We use time moving average to match the sampling frequency between the video principal components and the fMRI data. We employed a lasso regression because it offers both interpretability and generalizability. We ran lasso for each subject individually and store the beta coefficients for each principal component for each subject.
Right, so throughout this project, we found some roadblocks, mainly at first in setting up the work environment. So as I said, Ariel provided us an AWS VM machine like with 8 GPUs, so that was okay, but we also had to put all of the libraries inside and work out the incompatibilities. So after getting that done, like in day 2, we then determined the resampling sizes for the videos and the fMRI data, which was also well, for what we also had to do some hacking. Then we found a lot of errors running the slow fast model, first with the GPU drivers, then with the format of the data. So we were stuck there for a little bit too. And then our main roadblock was the differences between the training dataset that was used for training the slow fast model, which we used, and the HTTP dataset. So as Adriana will tell you later, you will see that the HTTP dataset includes very long videos, whereas the kinetics dataset used for training the network has very short videos which were oriented toward actions. So maybe we had to look into different datasets for this project.
All right, we're about to share some of our preliminary results. They're not exactly the most interpretable yet, but I think they serve as a fairly nice proof of concept for future directions. So for group level inference, there were multiple approaches we could have taken, but here we took the voxelwise beta weights and tested which were different from 0 on average across the participants. And here we're plotting the thresholded neuroimaging results from just 3 different principal components, along with the features that clustered within these components. So if you recall, all of our class labels from the slow fast algorithm were related to various actions and other kinds of biological movement. So probably the most interpretable result here is the component number 6, where we see a lot of voxels caring about the actions in the word cloud in occipital cortex extending laterally to like the superior temporal sulcus. And we also see some voxels in primary motor cortex that care about this kind of information, which is cool. And actually, one thing that I noticed during Shelby's slide was component number 1 actually shows a ton of more like complex and diverse actions, like tying a tie and giving or receiving a reward or playing the organ. And so I think it's pretty interesting that we see voxels in prefrontal cortex that care a lot about these particular actions, but very speculative.
Great, so what comes next? In terms of limitations, we have a few things that we'd like to address in the future. So as Alberto mentioned, since the slow fast weights we use were pre-trained on the kinetics data set, which primarily features physical actions, the labels are primarily reflective of physical actions rather than some of the kinds of categories that we were originally interested, like social versus non-social activity. That said, several of the HTTP videos, like the flower video we showed, don't really have a lot of physical actions involving people, so these labels were less useful in this context. And in addition, even though we have a decent amount of data per subject, we only use 30 subjects, as Sean mentioned earlier. In terms of next steps, in addition to organizing the repository that we have, we'd like to move away from using the pre-trained weights and try training slow fast on our data set, or on the HTTP data set, or on a more relevant data set. We'd also like to incorporate leave one out cross-validation at both the run and participant level, and then also apply our final approach on more subjects, or possibly on the Algonauts data set, which is a collection of 1,000 3-second videos that are more physical action oriented. And then lastly, we've discussed and actually started working on some alternative dimension reduction approaches, which Tiankang is going to talk us through.
Yeah, so for fun, we also trained an experimental LSTM model, which is a recurrent neural network. With this many-to-many recurrent neural network structure, it is now possible to perform encodings from both the fMRI and the video feature end, while preserving the temporal information and allowing for nonlinear interactions. We use 1D CNNs for video features and 3D convolutional neural nets for the fMRI data. The video feature encodings were input into the LSTM module to predict the fMRI encodings. The objective function was to minimize the Euclidean distance between the video encoding space and the fMRI encoding space.
Yes, and with that, that is the end of our presentation. Thank you so much. I think this is a nice time to thank NeuroHackademy for this opportunity, to thank all of our teammates for their brilliance and their hardworking late night hours, and also for the audience for just being a wonderful and wonderful, inspiring members of the NeuroHackademy community for the past 2 weeks. So thank you. And please check out our GitHub, which will be public later on after we clean our code. And if you have any questions, please shout out. Thank you.
That was incredible. Congratulations. Let's see what questions people have, comments.
So yeah, very impressive. I had a few questions and you answered them all on your future work slide. But I would just say as a comment, I've played around a little bit with not this kind of data, but with similar kinds of neural networks with brain data. And my experience has been that even if you start with an untrained or a pre-trained ResNet, that training on your data helps a ton. And that, in fact, I found that using an untrained ResNet to start with is even better. So I would be really surprised if you didn't see huge improvements when you did more training on your own data. But very impressive, cool project.
Yeah, actually, for that question, we are using like 3D neural networks, 3D ResNet. It's like we don't have like well-trained 3D pre-trained models there, so it was pretty sad. But are there like, I'm just curious, because like ResNet are trained, like ResNet trained on ImageNet are like 2D scenes, but are you aware of any like 3D scenes, like pre-trained 3D scenes?
Oh, that's a good question. I'm not, but I'm also not really an expert on this area. So
yeah,
there might be. There's a project called No Brainer that has some pre-trained networks for, I think it's for basically brain extraction. So that would work. Those are 3D networks for brain data. And I don't know if there are 3D in terms of like space and time networks that are pre-trained. Priya, you had a question? I saw your hand. But...
Not really a question. I just wanted to say, in addition to being technically and theoretically impressive, I also loved your use of memes.
Yeah.
The credit all goes to Sean.
Presentation. Great. Let's move on to the next presentation. Dicom parser. Oh, sorry, Waleed, I see your hand up before we go to Dicom parser.
It's okay. I just had a question. Actually, I'm completely, I don't have that much background in your project, but from my perspective, I just fail to, understand the point of PCA actually, and the map that you showed, which is, what is it called? Like the text map, or what is it called?
The word cloud.
The word cloud, which is, to me, actually is an overrepresentation of a specific, text, and then PCA is much more about variability within the data. And then I failed to connect between these two, concepts.
Yeah, so actually, we did this PCA to the video features because a lot of the words are overlapping with each other. For example, like showing a hammer or like showing a knife, So is that does anyone else want to like say more about why we do PCA?
Then this is the same. This is a feature I use for grouping between the different groups.
Yes.
The same. And then these are actually overrepresented in some specific group, basically.
Yeah,
that's why it's showing up. Yeah, I got the point now.
Yeah.
But your question leads to a really great discussion point that we constantly had over the nights, which was, we're sort of limiting ourselves in an emotion space where it's very, Slow Fast was particularly designed to extract action-related annotations. So we're limiting ourselves, and I think the next step that we discussed is we need to increase our variability and just swap the entire space instead of limiting ourselves to just motion annotations. So if we had something even akin to like object recognition versus action annotations, then we would have seen something more meaningful come out of the PCs. But just living in that action annotation space and applying PCs was probably not the best way to go, but just a thought experiment.
I see.
Yeah, to add on to Heejung's point, like we were thinking, yeah, maybe it might actually be most interpretable if we just fed the encoding model our full like feature by time or time by feature matrix, and then we'd find the voxels that care about all of these, you know, actions within this like within these like class labels that we've identified.
I see. Thank you. Thanks.
Great. Thanks for that clarification. Okay, Dicom parser. You're on.
Yeah. Thanks again. That last presentation was really awesome. I also kind of want that presentation theme, maybe for my entire desktop environment. I really loved it. In our project, Dicom parser kind of turned to Open Dicom. Finally, the repository which we were working on was originally under my personal account, and after some further discussions, which was a lot of our work in during this week, which I'm guessing is a part of it when you're collaborating on a project that's already kind of running to some extent. There's a lot of discussions to be had and a lot of decisions to be made. So that was a lot of that. And finally today, indeed, the repository was moved to the Open Dicom organization, and hopefully that will encourage collaboration in the future. And yeah, and I worked with Michal and Lee together. I think we each took on different aspects of the work, and it was really cool to see it combine. And I took this little quote from Wikipedia. I'm not a fan of a lot of text, but I thought it kind of summarizes the need for this package rather well. It says, while the Dicom standard has achieved a near universal level of acceptance among medical imaging equipment vendors and healthcare IT organizations, the standard has its limitations. Dicom is a standard directed at addressing technical interoperability issues in medical imaging. It is not a framework or architecture for achieving a useful clinical workflow. And I want to kind of replace the word clinical with scientific in general, I would say. And I just read that and I felt that it really resonated. I thought that was very well put and summarized the problem rather well. And so what the project pitch was based on the repo I was working on before, which is called Dicom parser, and it is essentially just as a reminder, a wrapper over PyDicom. And as a basis, it provided automatic conversion from Dicom value presentations to Python native types. And that was kind of the scope of it. And then I kind of started seeing how it could be useful to integrate some more of the functionality that was offered by NeuroHackademy and the CM2NIX, and then the community discussion kind of grew from there. And basically, now the working process is mostly on that part of adding more and more prior knowledge about vendor-specific information in order to kind of make sense into all those weirdly encoded pixel arrays and all the crazy metadata that is appended to those Dicom files. So yeah, that was most of I guess my work this week with parsing specifically multi-frame data, which is a kind of a newer style of encoding Dicom files. Unfortunately, it's not very widely adopted yet. I think it's getting more and more used around the world, and it kind of depends on with which vendor's equipment you're using, I guess. But I'm glad to say that that was completely integrated. And also, Dicom parser aims to provide the basis for higher level dataset abstractions. So, for example, mapping an entire Dicom archive to memory and facilitating access and manipulation. So if there's, I don't know, some hard drive that someone forgot in your lab with some Dicom files, and you kind of just want to quickly figure out what's in there, you could kind of throw that directory into Dicom parser, and it'll tell you, all right, 20 studies, 100 participants, that many scans. You can query by even perhaps sequence types and different header information like that, and maybe even manipulate the dataset so that it will organize automatically according to some format, or maybe even convert it to bids if we can get all that functionality done. So that's kind of the idea. And for the project, we kind of decided to go in the direction of documentation and community first, because that's really kind of lacking. We reorganized the documentation to be more accessible and easy to navigate. We created a resources page with many useful learning materials and other tools. I think I'll just kind of also show it. And we organized the differences in return types between PyDicom and Dicom parser so that a user can easily understand what those are exactly. I said about multi-frame support, and that includes 2 new classes that I really hope can also serve almost as documentation in a sense to that entire parsing process. That's kind of the beauty of Python, how it's almost like pseudo codish if you really abstract things enough. So I'm hoping that will be useful. And a lot of work it really has gone into just creating complete coverage for those new classes, because they're longer than I would care for, generally classes to be, but I kind of couldn't avoid that, and there was a lot of testing to be added for that. And on that note, there was a long discussion, both in the Discourse channel and a lot on Zoom, trying to figure out, what is the difference between DataLad and just having a few Git models. And I think between Michal and me, we kind of convinced the other person that the other method was the right way, and vice versa, like 5 times a day. But eventually we kind of, I think, settled on keep it simple, stupid, that general principle of just, if DataLad is basically Git models with some sauce, then maybe we don't need that sauce. Maybe our use case isn't that involved, and we should just rely on the simple and popular methods. So I kind of just want to go through the documentation quickly to just briefly show some changes we've done. First thing I think that was important was to make the quick start tutorial much shorter. It used to have a bunch of examples here that kind of cluttered the whole experience, and now it's just the image class, which tells you like how to use that class when you're reading single images, and the series class, which kind of summarizes how you would summarize header information from an entire series. And that's pretty much it, how to read the header information in pixel arrays. And that's it for the quick start. And then that's the table we added for all the different, for the typing between PyDicom and Dicom parsers. So just all those little changes, like between PyDicom's DA class and different things like that into the, Python native datetime date class, and etc. We moved the examples here so they kind of complement that information in a reasonable way, I think. Added documentation is fairly minimal, but I hope it achieves its purpose regarding multi-frame support, and kind of also improved a bit of the vendor-specific information. Currently, there's only Siemens, but there were little patch-ups to that. And finally, the resources page was also quite a bit of work, just summarizing a lot of good learning tools, and more importantly, just open source tools that are out there, most importantly, again, in Python, but also in JavaScript, C++, C, MATLAB, and finally Rust. So there's a good collection here. I personally even, I knew all those first 4 packages, but then going through all the discussions we had, I came across this Dicom 2Nifty, which we kind of mentioned, but then the contributors never really joined the discussion, and I never really dived into the code. And it turns out I was really missing. There's a lot of work that's been done there that I think could be super useful. And yeah, we might just integrate from there. So we'll see. So yeah, that's mostly the test files discussion, mostly conceptual. There's still a lot of work to be done. We kind of located all the files and made some order into that, but we still need to actually create the sub models within Dicom parser and edit the tests, which will probably not be that much fun, but also not too bad. Just changing a lot of fixtures. Yeah, that's pretty much it. Thanks to Michal and Lily and everyone for these great 2 weeks. And I'd be happy to answer any questions.
That's great. Thanks, V. Thanks, everyone. Questions, comments?
Nothing else. I'm glad to see that resources page because I feel like I end up explaining that to people all the time. It'll be great to have a link.
Yeah, I think the way that the documentation got a little simplified on one hand and also much more expanded on the other is just super. Yeah, I'll share later. Somebody shared with me this kind of framework for thinking about documentation, where you sort of split up your documentation based on the intended user audience. So different parts of that obviously have different audience, and you don't want to mix together the documentation that different people, a user or somebody who's new, or somebody wants to dive deeper, wants to have. So that's really thoughtful. Alberto, you have a question.
Yeah, I was going to say amazing work. It would be really cool if you were able to put together some really concise document on your workflows for simplifying the documentation, because you really did make a lot of progress.
Thank you very much. I really appreciate that. I'll give it some thought of how to abstract that. Yeah.
Great. Other questions, comments to the Dicom parser team? Okay. Let's move on to the overlapping networks team.
All right. Hopefully this works without too much trouble. Let me share the slides. Share that. Y'all can see that? Yeah. Yes, I know who I am. Thank you. All right. So this will be a little bit different than what we've been seeing for the other talks today. So we started this project as with the goal of assessing the overlapping network membership of network variants, but our presentation to you today will be more akin to many ways that you can not successfully do connectivity analyses. So I'll go ahead and pop over to the next slide. And
cool.
Yeah,
that's me. So quickly before we jump into the project or our many attempts to do this project, I just wanted to acknowledge everyone on this team. We are working with a number of people across many different time zones with varying familiarity with like SIFT files and also Python. So yeah, it's been a fun experience to work with everyone here, and a good opportunity to do team science.
Okay, so I can introduce what our initial project goals are, and then after that, we'll talk about how we did or didn't get to some of those ones. So the first one was to take these overlapping networks that Savannah had been working with in the HCP dataset and conduct overlapping network assignment at the group level with all of the Midnight Scan Club data subjects, and then compare those overlapping networks to the ones that are already have been found in the HCP data. The next thing was then because the Midnight Scan Club data, there's so many hours of data per person to really get those individual overlapping networks with each individual of the Midnight Scan Club data based on some work and thinking particularly about the network variant analyses that Seitzman et al. Had been doing with the Midnight Scan Club data to look at these really individual specific networks when we're looking at the overlapping networks. And then finally to look at these variants or places in the networks that tend to differ across the different participants, and really look at how the overlapping network membership, because we sort of from these algorithms that Savannah has been working with, we get a probability of each vertex or each area belonging to each network to see how overlapping network membership or membership in multiple networks or higher probability of membership in multiple networks might be related to status of part of the brain as a variant across participants or not. Oh, and then finally, to develop some norms for visualizing these overlapping networks, typically because it's a little bit tricky to show multiple networks if they have to overlap somehow or we have to blend colors. So you'll hear soon how some members of our group have really spearheaded an effort to try to come up with some new and original ways to visualize networks in this different way.
Yeah, so this is our hypothetical workflow. And this is just a pictorial representation of Paul has just discussed. And this is how we plan to move ahead temporarily as the time progresses. So initially we have planned to pre-process the data of individual subjects. And then based on that individual subject data, we plan to find out connectivity matrices as well as connectivity matrices for group averages of those subjects. And after we have we were supposed to have these connectivity matrices of group average and individual subjects, we were planning to calculate network variants, to perform network variant analysis based on the Stelmet's paper, and also the overlap assignment. And after this too, we can have this comparative analysis and also some norms to visualize this overlapping network. So this is what we planned. It was hypothetical initially, and let's see how it progresses.
Yeah, so what happened, right? We had these lofty goals. We want to walk you through our progress and our flow of issues that we ran into. So like we said, we wanted to at first compare connectivity matrices to be able to see comparisons between network variants and overlapping networks. But in order to make those connectivity matrices, we had to exceed the space possibilities of the Jupyter Hub. So that was the first issue. And then after that, we solved that by getting an AWS server. Thank you, Ariel and Noah, for getting that set up for us. Very helpful for a little bit. And then we ran into some other issues, right? So Midnight Scan Club data is, or the analysis pipeline is almost entirely written in MATLAB, not Python. So at first we were thinking we would maybe try to translate things into Python to really work within like the hub framework and in the spirit of NeuroHackademy. But then we were held up by the fact that we needed to, in order to get these connectivity matrices, which are otherwise known as Dicom files, we had to first convert time series files that were from the Midnight Scan Club data into these connectivity matrices. And the issue was that tools for making that conversion rely on this program called Connectome Workbench, which was also not available to us on the hub or the AWS server. And the SIFTI files from the Midnight Scan Club were not readable by Python imaging toolboxes. So, you know, a lot of science, I think a lot of computational science, you often run into this vortex like this. But, oh, and there's one more thing, I think. Yeah, we also ran out of time and space along the way, right? So these issues compounded on the fact that, you know, we didn't actually discover each issue until, you know, 24 hours later, because it took so much time to run one particular thing, blah, 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 right? So, you know, the hypothetical thing that would have happened is convert these SIFTI files into something that could be read by a Python imaging toolbox, and then convert that into a connectivity matrix, then convert those decon files into a representation of network variance, and then kind of test our hypothesis about overlapping networks and network variance and how those compare. So that top thing was kind of our goal. And we sort of, you can go forward to whoever's can join this, I think Savannah. Yeah, that top thing was our goal, and we sort of accomplished up to, that green box there. So maybe now we can dive in a little bit more into, our reflections on this process.
So just like Ben mentioned, at least with this project, we were able to figure out the bigger problem in the field of connectivity matrices. And we realized that a lot of the scripts that were already developed were developed in MATLAB. And we assumed that we could sort of use this and convert it into Python. And that led to us underestimating the time that it would take us to complete the project. And we were not able to estimate how complex the process would be. So one reflection from this project was also that given the relevance and the uniqueness of the MSC data, we think that future work should focus on translating the code into Python to make it more accessible to a wider audience. And on a general basis, the field of connectivity matrices could benefit from adapting a common language, say for example, Python, a standard file format, and maybe more efficient data structures and algorithms.
Yes, so while the others were struggling with the large data sets and long processing times and different data formats, I delved into a parallel universe with visualizations. So the idea was that by the time we have the results, we would like to be able to visualize them in an intuitive way. So my task was to prepare visualizations for this. And Savannah provided, based on an existing result, some matrices that I could use to play around with. And these contained 900 plus regions of interest, each row, and each column a network. And each cell would show which the probability of each region of interest being part of that network. Yes, and so in the end, we came up with several ways to visualize the networks. First, this image shows one single network and each region of interest, the probability of this region of interest belonging to this network. And this figure shows two networks, and the yellow parts show the overlapping regions between the two networks. And finally, we also thought it would be interesting to show the topography in a way that so how many networks each region of interest belongs to. So this is a single network, and all the connection the different regions have. So for the visualizations, I used NiLearn and NiPy, which was fun because I haven't used NiLearn before. So this was a learning opportunity for me.
All right, so it's a short but sweet presentation for us. So we'll just go over some of our conclusions and future directions. The first, which is very positive, is that we all know that you're successful at NeuroHackademy if you finish with a pretty picture of the brain. So check, done, we did the thing. The next thing that we took away from this project was that doing this at NeuroHackademy with everything compressed into a very short period of time, and everything being forced to be done all at once rather than the way we normally do our science, sort of stretching it out over weeks and weeks as we tackle one problem at a time. This really highlighted for us how many issues there actually are in working with connectivity data that otherwise might be obscured by, decay of memory over time. And it really told us that open data encode is really great. It's a fantastic thing to aim for, but it's also at the same time not enough to support the work that we want to do. First, we really need, especially for the connectivity field, we need tools that let us communicate across the platforms that we use and the analysis pipelines that have been developed. The size of connectivity data is just absolutely massive. I don't think that we actually referenced this yet, but one of our connectivity matrices for the HCP data set is in fact 33 gigabytes just for one person. And so this is a huge hindrance. Not every lab has the resources to have memory space for those sizes of data. And that really does need to be addressed. And finally, we need clear documentation on all of the different analyses and data types that we have. It's as important, if not more, than the sharing of the code and data itself. Finally, I would urge you to check out the visualization work that we have at our little mini package, NBpy, because we liked the name. We don't know what it stands for really, but that's fine. So that's what we have for y'all. Thanks everybody on my team who took my idea and just like totally ran with it. And thank you to Ariel and Noah and Jane for putting this fabulous workshop together for us. I think it's been a great experience for all of us, even if we don't have a fancy toolbox at the end of it. So thanks.
That's great. Really incredible effort there, and a lot of cool things that came out of this project. Tal, you have your hand up, I see.
Yeah, just a quick comment about open data is great, but not enough. I can't agree more, but I think that's where we come in. We need to look at opportunities like Dora presented, the BIDS extensions, or really any of the packages that we worked with offered the opportunity to contribute. And I've been exposed to that recently, and it's amazing. So bottom line, if there's something that you need and it doesn't exist, find a way to create it. And I think that what makes open source amazing, at least in my eyes.
Yeah, I should mention there is an effort to standardize these connectivity, the abstract representations of connectivity, like connectivity matrices and networks. I think it's led by Eugene Duff. It's a BIDS extension currently sort of in motion. And you know, if you see the gaps there, you should jump in and fill them in. I think, you know, the data sizes are going to continue to be very challenging, and it's hard, but it's also good, right? It's like you're dealing with really complicated systems at the scale at which they're complicated.
I'll just add that I feel very deep sympathy for all of the problems you encountered throughout this this project. I kind of feel like you experienced a little microcosm of what data science is often like in the wild, where you had you know, problems with weird file formats and code that couldn't actually convert it, and poor documentation and great big data sets, and all of these things happen all the time. And I kind of feel like a lot of times at NeuroHackademy we try to teach people how to walk through the minefield and what the path is, and you guys also learned where all the mines were, as well as learning the correct path. So props for that.
All right, other questions, comments? All right, let's move on to the last presentation by the Experiment Tracker team.
Hi. Can everybody see my screen?
Yes.
Okay, perfect. Cool. Okay. Okay. So we are the meta track. We shifted to a name that is easier to say. And so the goal of our package is to automate all the experiments log for your own sanity and to put it all in one place so that you can access it with ease. And I'm Haithvi. I'm David. And you'll notice that it's just two of us. Our
team has slowly been shrinking throughout the week, and it's just us two left. So we tried our best.
You really did. Okay, so today for you, we're going to outline what the package is going to be like, why it is required in the first place, what needs does it fulfill for an everyday researcher, break down the modularity of the different components, how somebody can access it in everyday life, how the setup has been, the different framework that has gone into the setup. Try to give you a short demo. We'll see. This is our first trial. We'll try. And then we'll end with questions, and you'll sort of see the problems we encounter on the way, and hopefully it's fun. Okay, so what is MetaTrack? So this is kind of David's brainchild, which I thoroughly support. And it's supposed to be a super tool to keep track of experiment data and metadata. So there's several key functions. Those include initializing experiments, a tracker to track all your runs and to call on the experiments that you have going on, a summarizer to summarize that experiment, any subjects, any data that you have, tracking the data, updating the data that you already have, and also tracking the progress on the overall project and the different modules that have gone into the project. So why do we need it? Experiments generate a lot of metadata. So even for one short everyday life, you have to keep track of how a figure was generated, or what went wrong with a particular scan, or why a participant isn't suddenly eligible, and what task version you used. But this list never ends because there's so much that we've just gotten so used to doing in our heads, but it's sometimes all over the paper or it's all over our digital landscape, and it's never in one place. So our goal is to consolidate all of it. So the three main needs that it fulfills is version control, but not just for your scripts. This is version control for your tasks, for the stimuli that you used, for the analyses that you conduct, and the figures that you generate. It also helps you keep notes on any issues that you might have had during the runs that you do, any subject attributes that you noticed, or any changes to the script that you had over the years. And it also helps you replicate your results better because it accounts for different code versions and directories. It also has a log for your figure parameters, so you can replicate those figures with ease. And it also accounts for explanations as to why you did an analysis in a particular way and all the different iterations for those analyses. So the modularity of the tracker, it has 5 main modules, and we've broken it down into modules so that the package runs faster and it's easier to get at different kinds of information. So the 5 modules are experiment, subject, stimuli, data, and writing. So as you will see, there are definite overlaps between these modules, such as each module has a name, a project name, subject names or IDs, stimuli set names, and so on. But each module also has unique variables. So just for writing, you can account for the collaborators that you have, different paper sections, where your citations lie, the journal requirements, pre-registration details. Whereas for stimuli, if you're creating your own stimuli set, you can have different versions for it, the number for each stimuli, any notes you have for it. And the one thing that's common across all of them is a progress summary and a progress bar, which you can access at any time to see how far you've come along and what needs to be done next.
So theoretically, once this is working, you can head over to our GitHub page. We're probably going to change it to MetaTrack pretty soon, but this is currently where it's pointing. But in theory, the setup is install Maria database or MySQL on your computer, and you can have that on a remote server. You can have that at home, and you can also have your meta tracker point to something remotely. And then you simply run the setup file, and then it should work. We've gotten it to work for us, but it doesn't work for everybody yet. So when we were organizing this project, there was a couple more of us, and we decided the best way to work on this was to split up the project into like its various like functions. And then so inputs from like the user, things that talk to other things that output data. And then we came up with like separate, I guess, parts to the overall project that we would work on independently. Next one. Under the hood, we have this database structure. It's pretty simple. I've changed it a little bit since, but the idea is to keep the database quite simple and to keep track of things that are mostly automatic, things that you could grab, like the date and time, the username of the person using the computer, the computer name, things like that, and be able to use this information in order to create like just a timeline of how your files are managed. Next one. Even further under the hood, we've got, when it came down to having our different parts work together, we decided to use this controller idea where you have the central hub that receives messages from the other units. And we use this subclass that we put on top of the units that will automatically connect with the controller on construction. And then this left part here, that is, we have some class that constructs, that is constructed from the database interface so that we can like do various things to the data.
So on the user end of things, we decided to start with a terminal interface because it can be accessed through most systems. And of course, in the future, we wanted to have a web interface and a GUI so that people can actually visualize their progress and see how all the data connects to each other. But for now, we use Click, which is a Python library to create our command line interface. And because we use Click, which is why a setup file is required. And so this is what our setup file looks like. It's a pretty basic file, but it basically just tells the terminal to look in the command file and then begin tracking all the user inputs and passing that on to the controller. So it's just an entry point for the terminal. The main chunk of user interface lies in our command file. And this is just a lot of information, but the summary of this is that for any program that we use for the terminal, there's lots of different options that we can type in for it to do different things. So over here, we're trying to create this exhaustive list of different options the user can input to either change the data in the database, to get the data from it, or to just input new information altogether. So a couple examples here that you can see is that there's a way for you to initialize your project name. And so if you just do -- project, it'll ask you for your project name and that's what you input. There's also ways for you to see how many subjects that you have left, how many subjects you have run. It accounts for how many total subjects you have, all your stimuli information. You can leave a note for your subject, for your run, for your version, for your stimuli, and all of this in the background gets connected to each other. And so even when you were to pull information, a lot of these commands you just leave blank and that will output information from the controller. And so of course, this list is not exhaustive yet, but these are just like 20 basic options that we have in place as of now, and we'll keep working on it. And okay. I'm not sure if this part is going to work.
You have to stop sharing and then I can.
Okay, yes. Let me stop sharing. There we go. Sorry, we did have to switch laptops for this part.
So here I'm at, let me make the.
David, this is really small. If you could make it much, much bigger. Yeah, there we go. That's good.
Okay, so let me move this up here just to some of this. Okay, so if we had like one more day or even like 3 more hours, this would work a lot better. Like here would be an example of making me like a query through this. This Python 3 would be gone when we're getting this to work. So you just run meta-track. And then it takes in like arguments, like this would be like the name of my object, this would be its type, and then this would be where it's located. And then I could insert it into the database if insertions were working right now. You can see like that it's connecting to the database. If I turn on some debug here, like you can see that like I'm connecting to the database, and there's some inter-process communication between the various parts here. So here's like units talking to the controller. Let's see, I think that's all I was able to get working right now, but it's a work in progress. There's a lot of code that's come into this so far, and it's just like now barely coming together.
Yes. And also yesterday after I debugged Click with Ariel and Eric, suddenly we had a clear path, and I had to do so much coding yesterday. But so now we're on a good path. So honestly, one more week and we would have a functioning package. Okay, let me share my slides again. That is cool. So we already spoke about our roadblocks, but at this point, if anybody has any questions about the package, anything we can help you out with, anything you think that we should even include that we haven't yet, so any comments that you have, we'd love to hear them.
That's great. Questions, comments to Team MetaTrack?
I have a question.
Oh, go ahead.
Sorry. So yeah, this is wonderful. And I think we all sort of like have experienced this problem. Like there's so many metadata that you need to keep track of. And the OSF.io and probably like lots of platforms out there have this. Have you ever thought of sort of providing features where you can integrate it with different platforms?
We wanted to build it flexible enough that you could do that pretty easily. Right now, it's more like just prototyping, but eventually that would be very nice. It'd be nice to be the one who solves science with a tool, you know. But,
yeah, I'd be interested in using that. Thank you.
Yep.
Thank you.
Yeah, this is really cool, and I've spent many years complaining that no one has built this tool yet, essentially. So I'm glad to see people working on it, and really excited to try it out. Just a quick suggestion. Given that you have like a database running in the background, a nice feature would be eventually if you wrap some of that up in a Docker compose file, so that it was very easy to just have it start and not worry about some of the tricky details like installing a database and that kind of stuff.
That's a good point.
Yeah, if you're interested in doing that, I'd be happy to help you with that.
Yeah, definitely. That might be even good for us when we get to testing. So, or
Yep.
just early on even. So yeah, we'll talk to you about that. That's awesome.
Well, thank you.
Other questions, comments here? All right, I think we have reached the end of these presentations. So first of all, a big round of applause to all of you for all the amazing presentations and all this work that you've done over the course of the last few days. It's been really amazing. Again, really amazing to see. And then just a few words of summary here. One is, please don't forget to fill out the survey that we sent on Slack. Few more things. First of all, Sparkle is going to be available for another week or so. But then I've been asked, the videos that are on Sparkle in the Pariah library will also be available on YouTube to view after that 1 week, for anyone to view. And they'll be linked from the NeuroHackademy website. So if you go to the course year for 2021 and then click through the titles of the different presentations there, including this set of presentations that you just made yourselves. The videos will be available there for you to view. And then just a request to stay in touch on Slack or otherwise. If you're doing stuff that you want to share with us, we'd really love to hear about how these projects evolve into your own work, and how the things that you've learned over the course of the last 2 weeks become part of things that you do when you after you've had the opportunity to rest for a little bit and go back to all the things, all the wonderful things that you all are doing. And with that, I think I will close. I hope to see you all soon in some way or another at conferences or at other opportunities, and wish you good luck in everything that you do. Have a nice weekend, a restful weekend, I hope, and see you all soon. Take care.